to each other. So in other words, the R groups are not involved in any of this hydrogen bonding that forms the secondary structures like the alpha helix. It's just hydrogen bonding from the backbone. And the same is true with the beta sheet secondary structure. That's the zigzag back and forth pattern. Here it is close up. The pink again is the backbone. It zigzags back and forth. And again, you can see it's just the backbone hydrogen bonding that's driving it. The R groups are not involved in any of the secondary structure folding. All right. So now let's imagine you're just going to let go of the ends of the protein. Let go, boing. Just let it fold however it wants to fold. Well, what will happen then is it would fold itself up even more. In other words, this beta sheet and this alpha helix would sort of fold together like that, and that's called tertiary structure. Tertiary is the folding together of the secondary structures. And the interactions of the R groups are what drives tertiary structure. And notice that's different. When we were talking about the secondary structure, the alpha helixes and the beta sheets, that was not the R groups, that was the backbone. For tertiary structure, it is the R groups and not the backbone that drives that folding. And here's a diagram showing that. The blue ribbon there is the backbone, but you can see some R groups sticking off the backbone there. There are several different types of interactions between the R groups that drive tertiary structure folding. One is hydrophobic interactions. If you have two hydrophobic R groups, even if they're on different parts of the backbone, like there's an amino acid here, and there's an amino acid up there, those hydrophobic R groups don't want to stick out into the watery environment because they're hydrophobic. They want to cluster together in the interior of the folded protein where they can escape from the watery environment. So the hydrophobic R groups seek out each other so they can clump together to escape from the water, and that tends to fold the protein out. Other R groups can have ionic charges, and if you have a negative ion R group, it will be attracted to a positive ionic R group, and that will tend to fold the protein up like that. So you get ionic interactions. Even R groups that aren't ions but have partial charges can get involved in tertiary structure. Here's a partially negative oxygen, partially positive hydrogen there. They're going to hydrogen bond to each other and cause some tertiary folding. And what I think is the strangest of all, some R groups have a sulfur atom as part of their structure. And if you get two sulfur atoms near each other, they can spontaneously form a new covalent bond. It's called a disulfide bridge, and that can pull together two amino acids that are on different parts of the protein, and that, again, that causes some folding to go on. An interesting little tangent thought about this. You've probably heard of people getting a perm for their hair, where it makes their hair curly. When a person gets a perm, the hair is made out of a keratin protein. What they do is they add a type of substance that breaks these disulfide bonds to the person's hair. And so that allows the proteins of the person's hair to relax their shape. And then they wrap the person's hair around curlers and then restore these disulfide bonds. But the proteins then get locked into this curly shape. And well, that's what causes them to have the permanent wave in their hair. And it stinks also because sulfur is just kind of an intrinsically stinky molecule. Okay, so there's the tertiary structure, the folding together of the secondary structures. Any questions up to this point?
Do you know the part where you showed the slide where you're holding up to
Let me back up here for a second.
I think it's that one.
Yes.
So you know that right there you're talking about the alpha and the beta?
Uh-huh.
And you said, like, pretend that you're pulling it, right? And I'm asking, like, in the natural world, does it actually happen that it just pools by itself?
No.
Or is it just us?
That's how I'm introducing the topic.
Okay.
And I'm glad you mentioned, it's something I was gonna mention here. Let me get back to, not that
So
slide.
it'll never look like.
It would never be straightened out.
Okay.
No, okay. Here we go. Yeah, okay, so yeah, I'm glad you mentioned that because I only use that as kind of a way to introduce the topic. In real life, proteins are never pulled straight like that. In other words, all three of these levels that I've talked about of folding exist simultaneously. Like this protein right here, it still has primary structure. It has methionine, glutamine, alanine, methionine, aspartic acid. It still has a primary structure. It still has a secondary structure. Here's some alpha helix. Here's some beta sheet right there. And it still has tertiary structure, the way those secondary structures are folded together like that. And so really, in real life, all structures are present simultaneously. And in my example here, I showed this protein just having one alpha helix and one beta sheet, and it could certainly have more than one. Several sections could be alpha helixes, and several sections could be beta sheets, that's fine. Okay, now there's one more level of folding, quaternary structure. And you might say, well, how is that possible? We already let go of the protein. It's already folded itself up. What other level of folding could there be? Well, remember that some proteins are made out of more than one polypeptide chain folding together. And if that's the type of protein you're talking about, you can talk about how the two different polypeptide chains fold together. And that's what quaternary structure is. If this protein has a second polypeptide chain, how they click together with each other is the quaternary structure of the protein. Can a single polypeptide protein have quaternary structure? No, because by definition quaternary is only for proteins that have more than one polypeptide chain clicking together. And this is another good example of what you asked about earlier. Even in a protein with quaternary structure, all structures are there simultaneously, primary and secondary and tertiary and quaternary, all going on at the same time. Okay, now a slight bit of subject change here. I want to talk a little bit about the functioning of proteins, and I'll use an enzyme as an example. That's not an enzyme, it's hemoglobin. Why did I put that up there? Oh, just this hemoglobin is an example of a protein with quaternary structure because it is four polypeptide chains all folded together. Okay, now this thing in blue here is supposed to be an enzyme. I didn't make it as colorful as I sometimes do, but it is an enzyme. And just for a quick review here, remember that enzymes have an area where they bind to their substrate. The substrate is the molecule that the enzyme works with. And so all enzymes have an area called the active site, which is the crevice where they bind to the substrate. The substrate clicks into the active site. The enzyme then carries out some sort of chemical reaction on the substrate molecule, changes it in some way. In this particular reaction, this enzyme is breaking the substrate into two smaller product molecules. Other enzymes might do a different reaction. Other enzymes might join together smaller molecules to make a larger molecule. But anyway, this is the enzyme that we're talking about. Oh, here's a review question. What type of structure do you see here? That's supposed to be a beta sheet. And here is supposed to be a alpha helix. Some areas of the protein might not have any secondary structure. Like this zone right here doesn't appear to be either an alpha helix or a beta sheet, and that's fine. You can have areas with very little secondary structure. Anyway, so here's this enzyme carrying out its particular chemical reaction. And when they're talking about enzymes, they sometimes talk about the rate of the enzyme. And that just essentially means how fast the enzyme is carrying out its particular chemical reaction. And usually you can measure the rate in how fast it's producing its product molecules. That's a good way to measure a rate. The temperature of the enzyme influences what its rate is. Here's a graph showing for a particular enzyme the rate on the y-axis versus the temperature here on the x-axis. And so notice that there's a certain temperature that's optimum for this particular enzyme where it has its fastest rate. But if you go colder than that, the rate gets lower. And if you go warmer than its optimum temperature, the rate gets lower. Now, how is it that the temperature controls the rate of the enzyme? Well, if we begin here at the lower temperature, it just comes down to simply that at colder temperatures, molecules are moving more slowly. Here's the same enzyme in ice water. All molecules move slower at slower temperatures, and so the substrate drifts in more slowly, and then the enzyme does its chemical reaction more slowly on the substrate, and the products leave more slowly. So the whole thing is just going to be a little bit slower the lower temperature you put the enzyme in. If we crank up the temperature a little bit to something around room temperature, then since it's warmer, the molecules move faster, and so the rate will increase as we increase the temperature. And if we crank up the temperature a little more to body temperature, it's going to go even faster. So far so good. We're explaining this part right here, why it gets faster as you get warmer. But what seems a little bit weird at first is why suddenly does the rate drop off? You know, you'd expect from what I said earlier that the higher the temperature, the faster the molecules will go, so it should just keep getting faster and faster. Well, the drop off occurs at higher temperatures because of the enzyme unfolding. If you put this enzyme in boiling water, for example, there's so much kinetic energy. The molecules are moving so quickly that the enzyme ends up getting unfolded. You know, it's getting pounded by water molecules moving at a zillion miles an hour. Well, not a zillion miles an hour. Actually, I just read something about this. Around 400 meters per second is what water molecules will be moving at. You don't have to know that number, but that's how fast they would move at 100 degrees Celsius. And plus the enzyme itself has a lot of kinetic energy at that temperature, and so the hydrogen bonds that are keeping it folded would tend to be broken pretty easily. And anyway, it would unfold itself. Goodbye, active site. And that process is called denaturation, where you make a protein like an enzyme inactive because you've changed its shape, because you've unfolded it. And pretty much always